Welcome to AP Biology, Unit 1. Let's get started. First, we're going to cover the properties of water. There are 5 properties of water that you need to know for the AP exam. First is adhesion. Adhesion is water's ability to be attracted to something other than itself, and this is caused by hydrogen bonding. The next is water's ability to act as a solvent. So water can dissolve ions and other polar substances. The third is cohesion, which is water's ability to be attracted to itself and to hold together. This gives it surface tension. The fourth is density as a solid. Water is less dense in its solid form, which is not a common trait shared by other liquids. This is why ice will float on top of the water rather than sink to the bottom. And the fifth is water's high heat of evaporation. It requires a lot of energy to break those hydrogen bonds and turn a liquid water into gas. The combining of cohesion and adhesion gives water something called capillary action, which is water's ability to climb up things and move essentially against gravity, because it's forming bonds with one thing and it's allowing it to climb up by forming bonds and releasing bonds in order to scale a wall or a side of a beaker or a paper towel. Going forward, these are some basic units that you need to know in order to be successful in AP Bio. First, an isomer is 2 molecules with the same formula but a different structure, like these ones here. These isomers are also a mirrored image of each other. Often one is effective in a reaction and the other is not effective in a reaction. The term monomer is used to describe a molecule viewed as a single unit, where a polymer is multiple monomers strung together in a chain. So monomer is 1 unit, polymer is multiple units put together. And multiple polymers put together is called a macromolecule. Anabolic reactions build polymers, and catabolic reactions break monomers. In order to understand how polymers and monomers are built, you need to understand dehydration synthesis. Dehydration synthesis is the attaching of 2 monomers, and when these monomers attach to each other, they secrete H2O as a byproduct. Next is hydrolysis. Hydrolysis is the process in which polymers break into monomers. You can think of hydrolysis as the reversal of dehydration synthesis, in the sense where if you add water, the polymer will break apart into monomers. There are 4 major classes of biological macromolecules that you need to know: proteins, carbohydrates, nucleic acids, and lipids. Let's look at each of them individually. First is carbohydrates. Carbohydrates are composed of carbon, hydrogen, and oxygen. This includes things like sugar, starch, and cellulose. Carbohydrates supply energy for the cells, form plant structures, and work as short-term energy storage. There are 3 types of carbohydrates that you need to know. First is cellulose, which is structural material of plants. It's very sturdy and arranged linearly, like this. Second is starch. This is how plants store energy. It's weakly arranged, so it doesn't make as good sturdy structures as cellulose. Finally is glycogen. This is how animals store their energy. It's more sturdy than starch, but less sturdy than cellulose. Next up, lipids. Lipids are comprised of carbon, oxygen, hydrogen, and phosphorus. Lipids are most often used to store large amounts of energy and to form boundaries around cells known as membranes. What you need to know about lipids is specifically triglycerides. There is saturated and unsaturated triglycerides. You've probably heard of this as saturated fat versus unsaturated fat. Saturated fat has hydrogen. It's found in animal fats, it has a straight structure, and it is a solid at room temperature. Unsaturated is double bonded carbon. It's found in plant oils, and it has a bent structure. This is a liquid at room temperature. It is important to note that both saturated and unsaturated fats are nonpolar, so they do not dissolve in water. Your blood, which is mostly made of water, cannot dissolve these fats. Lipids are most useful when talking about the phospholipid bilayer, which is a membrane that all of our cells have. A phospholipid is made of two fatty acid chains, a glycerol and a phosphate. This is responsible for the membrane structure. Phospholipids have a hydrophilic head and a hydrophobic tail. The heads are going to be pointed to the outside because they can interact with water. The tails are going to be pointed to the inside because they cannot interact with water. And there are two layers because the head is going to also interact with the moisture inside of your cell as well. Next is proteins. Proteins are comprised of carbon, oxygen, hydrogen, nitrogen, and sulfur. Proteins regulate the cell process and build structures. Some examples are enzymes, hair, and skin. All proteins are identical except for the R group. This is how we determine which protein we're looking at. You need to know the 4 steps to structuring a protein. The first is the primary structure. This is linear and it looks like this. The secondary structure, which looks like this, is formed with hydrogen bonds. The tertiary structure, which looks like this, is caused by the interactions between the R groups. R groups can be hydrophobic and nonpolar, hydrophilic and polar, and have a positive or negative charge. For example, a positively charged R group is going to attract a negatively charged R group. A polar R group is hydrophilic, so that's going to be pushed to the outside. A hydrophobic R group is pushed to the inside. A disulfide bridge is the connection of 2 R groups both containing sulfur. Polar amino acids have a lot of oxygen and nitrogen, where nonpolar have overwhelmingly carbon and hydrogen. And the fourth structure is caused when 2 or more of these tertiary structures are linked together. Finally is nucleic acids. Nucleic acids store and transmit genetic information. These are composed of monomers called nucleotides. A nucleotide structure has a negative charge and is polar. The structure is comprised of 1 phosphate group, 1 sugar, and 1 nitrogenous base. The nitrogenous bases for RNA versus DNA are different. Let's talk about the differences between DNA and RNA. DNA is a double helix structure. It is antiparallel and moves from the 3' to 5' direction. DNA's nitrogenous bases are comprised of A, T, G, and C, all of which stand for these. A always bonds with T, and G always bonds with C, and vice versa. It's also interesting to note that G and C have a stronger bond because it has 3 covalent bonds, not 2. The bond between A and T only has 2 covalent bonds. RNA is a single-stranded piece of genetic information. Instead of thymine or T, it has uracil, which is represented as U. Unlike DNA, which has deoxyribose as a sugar, RNA is constructed with ribose as a sugar. DNA has 2 jobs. First is to replicate exact copies of itself. Second is to carry the instructions on how to make proteins using RNA.